So, today's session we are starting nerve muscle physiology. So in this session we will learn about neurons and neuroglia. So nerve muscle physiology is having many topics. First we will try to understand the structure and the functions of neuroglial cells and about the neurons. Then we will go into the detailed topics one by one. So neuron is the structural and functional unit of the nervous system, which is actually a basic building block of the nervous system. There are 100 billions of neurons in the human central nervous system. And more than these neurons, there are supporting cells which are called as neuroglial cells, which is about 10 to 50 times more than the neurons. So there are 100 billions of neurons. 10 to 50 times more than these neurons, there are supporting cells which are called as neuroglial cells. And these neurons, they have three basic parts, which is called a cell body, and this is axon, this is dendrites. So this cell body, which is also called as soma or perikaryon, or it is also called as pteridodendria. So cell body is also called as soma, perikaryon and telodendria. And here you can see processes which are, these are the small processes which are called as dendrites, and there is one long process which is called as axon. So this is the typical structure of the spinal motor neuron, which is actually a multipolar neuron. I'll tell you what is multipolar neuron. So here you can see there are different many poles from which dendrites and axon arises. So this type of neuron is called as multipolar neurons. So this is the typical structure which is seen in the spinal motor neurons. And two important terms we must know, that is nucleus and the other one is ganglion. So these cell bodies, the collection of cell bodies within the central nervous system, it is called as nucleus, and the collection of cell bodies outside the central nervous system, it is called ganglion. While studying about central nervous system, we are going to learn many nucleus. For example, I'll tell you one nucleus. When we study about hypothalamus, we are going to study about suprachiasmatic nucleus, which is the biological clock which actually regulates the circadian rhythm of the body. So so you know, that is the nucleus, which is suprachiasmatic nucleus. What is meant by nucleus? It is nothing but the collection of cell bodies within the central nervous system. And ganglion, which means the collection of cell bodies outside the central nervous system. And here we can see the structure of the cell body. So cell body is, it may be many in shapes. It may be in different forms of shapes, like it may be round, it may be stellate, or it may be pyramidal, or even fusiform in shape. It is the one which contains nucleus. So this is also called as metabolic center of the cell. So and the cell body contains all the cell organelles. Similarly, like cell, it is also going to have all the cell organelles like ribosomes and endoplasmic reticulum. It is going to have mitochondria. It is going to have all the other cell organelles, except that it doesn't have the centrosomes. So they lost the ability of cell division. So this neurons, if they are destroyed, they'll be replaced by neuroglial cells only. So this cell body, you can see there are some granules in the cell body. So these granules are called as Nissl granules. When we studied about smooth rough endoplasmic reticulum, I told you the rough endoplasmic reticulum is present abundantly in the Nissl granules of neurons. So here you can see the cell body of the neurons contains Nissl granules, which is actually containing more number of rough endoplasmic reticulum. So it is the protein machinery which is for the neuronal cells. So this is the place where the proteins and neurotransmitters are synthesized, and they are transported to the nerve terminals, and the transport mechanism is called as axoplasmic flow, where the axoplasm is the cytoplasm of the axon. So the substances which are synthesized by the cell body are transported by axoplasmic flow, and where it will be there within the nerve terminals. This is the terminal portion of the axon, which is called as axon terminal or nerve terminal or terminal button or the synaptic button. So here these synthesized proteins or neurotransmitters, they will be stored in the form of, they will be stored within the secretory vesicles. So these nerve terminals, they will contain secretory vesicles, but the neurotransmitter and proteins are synthesized from the cell body, because here is the protein machinery. So in addition to this Nissl granules, the cell body also contains neurofibrillae and pigment granules. This neurofibrillae contains microfilaments and microtubules, and in some neurodegenerative diseases this will degenerate. These neurofibril filaments will be degenerated, and there in some neurons, especially we can say one example where the neurons which is present in the substantia nigra, they'll contain a pigment which is called as neuromelanin. So in some neurons there are pigment granules, for example neuromelanin, which is present in the substantia nigra. So here we can see the structure clearly. So here we have Nissl granules, which is the protein machinery. I told you this Nissl granules are not present in the dendrites as well as in the axon hillock. What is this axon hillock? Axon hillocks is the thickened portion of the cell body, and this is the place where there is no Nissl granules, and even in the dendrites there is no Nissl granules. Nissl granules are present only in the cytoplasm of the, that is the cell body, the cytoplasm of the cell body. And these Nissl granules, they disintegrate and they form like a fine dust structures when there is degeneration of the neurons. And this process, the degeneration, disintegration of these Nissl granules in the form of fine dust and disappearance of these Nissl granules during degeneration of neurons is called as chromatolysis. That is a process which occurs during degeneration of the neurons. And I told you neurofibrillae, which is microfilaments and microtubules, which alters in neurodegenerative disorders, and the pigment can be present in the cell body, and neuromelanin, which is present in the substantia nigra. That is the example for this pigment. And the dendrites. So dendrites are multiple short processes. Here you can see there are short processes which are arising, which are extending out from the cell body, and there are in some neurons, these dendrites will have small knobby neck projections which are called as dendritic spines. Especially these dendritic spines are present in the neurons which are present in the cerebral cortex and in the cerebellum, and this is the receptive process, which means this dendrites can synapse with the other neurons, and they can receive the signals from the other neurons. So this is the receptive process, and this is the place where graded potential is generated or local potential is generated. So in the dendrites, the graded potential or the local potential is generated in the dendrites. And here we can see the cell body. It is the, so from dendrites the impulses are transmitted towards the cell body, but from the cell body the impulses are transmitted away from the cell body through the axon. So it is this is the one which is conducting the impulses to the nerve terminals. So axon. So we told there is only one long process which is called as axon. So in this, I told you the axon hillock is the place where the thickening of the cell body. So it is a thickened portion of cell body is the axon hillock, where this from the axon hillock this axon arises, and the first portion of the axon is called as initial segment of axon. So the first portion of axon is called as initial segment of axon. You can see the axon is lined by myelin sheath. Axon is lined by myelin sheath, and this this myelin sheath is not continuous. In some places there is absence of myelin sheath, which is called as node of Ranvier. Node of Ranvier is the place where the myelin sheath is absent, and this axon it ends as the axon loses its myelin sheath and divides into a number of fine branches, and they end as small swellings which are called as terminal buttons or terminal boutons, or synaptic knobs, which is also called as nerve terminals. So all these are other names for terminal button, and this is the place where I told you the neurotransmitter or the proteins which is synthesized in the cell body, they'll be transported by axoplasmic flow and they'll be stored in the form of secretory vesicles in these nerve terminals. So this myelin sheath, about this myelin sheath. So the myelin sheath is formed by the type of neuroglial cells which are called as macroglial cells. There are three different types of macroglial cells. Neuroglial cells are basically two different types, microglial and macroglial cells. Neuroglial cells are only 20%, whereas the macroglial cells are 80%. Neuroglial cells are mainly the scavenger cells. They are mainly phagocytic cells in the central nervous system, whereas the macroglial cells, there are three different types of macroglial cells. One is the oligodendrocytes. The other one is Schwann cells. There is a third type of macroglial cell which is called as astrocytes. There are two different type of astrocytes. We will study detail in the later slides. Just remember, the macroglial cell is a type of neuroglial cell, which are the supporting cells, and this macroglial cells have three different types of cells. The two cells are coming here, which is oligodendrocytes and Schwann cells. This oligodendrocyte cells, they form the myelin sheath in the neurons, which is in the central nervous system, and the Schwann cells are the cells which are forming the myelin sheath in the peripheral nervous system. So oligodendrocytes forms myelin sheath in the central nervous system, whereas Schwann cells forms myelin sheath in the peripheral nervous system. So these Schwann cells, they wrap around the axon for 100 times, and this Schwann cells are compacted by a protein which is called as zero protein. So there is a protein which helps in the compaction of the myelin sheath, which is called as protein 0. And in some neurodegenerative, some autoimmune disease, multiple sclerosis is an autoimmune type of disease where the myelin sheath can destroy, and there can be conduction blocks in the neurons. And this you can see node of Ranvier, right? So node of Ranvier is present at where there is absence of myelin sheath, and it is present for 1 micrometer for every 1 millimeter length of the axon. So to know more about myelin sheath is actually a protein lipid complex where there is more of lipids, 80% of lipids and only 20% of. So if we take protein lipid ratio, protein is 20% and the lipid is 80%. And you know that myelin sheath is an insulator where it will block the conduction of impulses. So the impulses will be jump from one node of Ranvier to the other node of Ranvier, and that is a type of conduction which is called as saltatory conduction, where we can't say that all the neurons are myelinated, all the axons are myelinated. In some neurons, the axons are not myelinated. They are called as non-myelinated axons. So the axon is myelinated, or it can be myelinated axon, or it can be unmyelinated axon. In myelinated axon, the conduction will be faster because the signals or impulses will jump from one node of Ranvier to the other node of Ranvier. So this type of conduction is called as saltatory conduction. So if the myelin sheath is not present, for example, I told you the multiple sclerosis disease where there will be conduction block. So if there is no myelin sheath, what will happen? If the myelin sheath is destroyed, the conduction will not be faster. The conduction block will occur. So to summarize the parts of neuron, so cell body, I told you the other name for cell body, soma, pericardia and telodendria, and it is the one which contains nucleus, and it is the metabolic center. And the dendrites are the small processes which receive information from other neurons, and it has dendritic spines, especially in the neurons which is present in the cerebral cortex and cerebellum. They will have some small knobby like projections which are called as dendritic spines, and axon hillock, it is the thickened region of the cell body, and axon is the long process which transmit signals away from the cell body to the nerve terminals. Initial segment is the site where initial segment is the first portion of axon. It is the place where the propagated action potentials are generated. So in dendrites, the graded potentials are generated, whereas in initial segment, propagation, which means the impulses can be propagated. So propagated type of action potentials are generated in the initial segment. And nerve endings is the site where neurotransmitter is stored, and it can be released from nerve terminal to the next effector organ, for example muscle. And to come to the different types of neurons, I told you spinal motor neuron, which we have seen in the first slide, which is a typical structure which is showing the neuronal parts. So spinal motor neuron. Here we can see there are different types of neurons. It is a multipolar neuron. So there are, based on the number of poles, we can classify it into unipolar neuron, bipolar neuron and multipolar neuron, and depending upon the function, neurons can be classified into sensory neurons and motor neurons. So what is meant by sensory neurons? These are the neurons which carry sensation or the sensory impulses. These are the afferent nerves which carries impulses from the periphery to the center, whereas the motor neurons, these are the neurons which brings the nerve impulses from the center to the effector organ. For example, it may be muscle or it may be gland. So depending upon the length of the axon, the neurons are classified into Golgi type 1 neurons and Golgi type 2 neurons, where the length of axon may be short or long. So here you can see a picture which is showing different types of neurons based on the number of poles, right? So here you can see there is a cell body and there is only one pole from which the axon and dendrites arises. So this type of neuron is called as unipolar neuron. It is mainly present in invertebrates. So unipolar neuron is mainly seen in the invertebrates, and the bipolar cells, which is especially seen in the retina, and even they are present in the olfactory epithelium. So this was the MCQ which was asked in the previous year question. So bipolar neuron is present in the retina as well as in the olfactory epithelium. So here you can see there is a cell body from which there are two poles. From one pole axon is arising, the other pole dendrites are arising, and this is the subtype of bipolar neuron, which is a type of sensory neuron. So from here you can see cell body. This is a type which is called a pseudo unipolar neuron. Initially the cell body will have only one pole, which is like unipolar neuron, right? But this is divides into two, and during development from this process, two axonal endings will arise, and one will go to the spinal cord, the other one will go to the skin or to the muscle. So this both of these are axons. So this axon, you can see, this is the central axon which is going to the spinal cord. This is the peripheral axon which is going to the skin or to the muscle. So from cell body, initially there will be only one pole, which is then divides into two poles of axons. So that is why this is called a pseudo unipolar neuron. So during development, this single pole will form two poles, and so this is a subtype of bipolar neuron. So this is especially seen in the ganglion cells of dorsal root ganglion. The dorsal root ganglion cells will contain this type of pseudo unipolar neurons. And all these are the examples for multipolar neurons. You can see this is spinal motor neuron, which we have seen in the earlier slide, and this is the pyramidal cells which is present in the hippocampus, and this is the Purkinje cells which is present in the cerebellum. So all these are multipolar neurons. So you can see there are multiple poles from which the dendrite and axon arises. So these are the types of neurons which is based on the number of poles. So this is the summary to classify the neurons. So here we have unipolar neuron, which is especially seen in the invertebrate neurons. There will be single process which is originating from the cell body, and from where from single pole both axon and dendrite arises. Bipolar neuron, which is seen in the retina as well as in the olfactory epithelium. So there are two poles from which axon and dendrite arises. Pseudo unipolar neuron, it is a variant of bipolar cell, which is seen in the dorsal root ganglion cells. Multipolar neuron, there will be single axon but there will be many dendrites, and this is seen in the spinal motor neuron as well as in the pyramidal cells of hippocampus and Purkinje cells of cerebellum. There are many multipolar neurons which is present in the central nervous system. So here we can see a picture which is showing different zones of neurons. I told you this is dendrites, which is the receptive process. So here is the place where graded potential is generated, and this is the cell body, and in the initial segment of the neuron there propagated type of action potentials are generated, and this is the place where axon nerve impulses are transmitted to the nerve terminal. So this is the propagated zone, and this is the zone which secretes neurotransmitter. So this is the area where zone of secretion of synaptic transmitter. This is the nerve endings. So these are the different zones of neurons, and the site of propagated action potential generated different for motor neurons as well as for sensory neurons. So in spinal motor neurons, initial segment is the site of generation of propagated action potentials, and in cutaneous sensory neurons, first node of Ranvier, initial node of Ranvier or first node of Ranvier is the site of generation of propagated action potential. So we should remember what the two things. One is the graded or local potentials are generated in the dendrites, whereas the propagated action potentials are generated in the axon. In spinal motor neurons, initial segment is the site, whereas in cutaneous sensory neurons, first node of Ranvier is the site of generation of propagated action potential. So this can be a MCQ question which can be asked. And coming to the neuroglial cells, I told you there are two different types of neuroglial cells are supporting cells in the central nervous system. I told you in the first slide that neurons are 100 billions, but more than neurons, 110 to 50 times the neuroglial cells, which are supporting cells, are present in the central nervous system. So this neuroglial cells are two types: microglial cells, and which are 20%, and macroglial cells, which are 80%. So here we can see a clear picture which is showing these are oligodendrocytes. I told you oligodendrocytes, Schwann cells, and astrocytes. These are the different types of macroglial cells. So macroglial cells are three different types: oligodendrocytes, Schwann cells, and astrocytes. Oligodendrocytes, they form myelin sheath within the central nervous system. So they form myelin sheath within the central nervous system. One important point we should remember is these oligodendrocytes, this single oligodendrocyte, so you can see it forms myelination in many neurons. It forms myelination in many neurons. Actually, the ratio of myelin sheath formation by oligodendrocyte is 1:30. So one oligodendrocyte can form myelin sheath around 30 neurons, whereas in case of Schwann cells, the ratio is only 1:1. So one Schwann cell can form myelination in only one neuron. So the ratio here is 1:1, and the ratio here in the oligodendrocyte, it is 1:30. You can see in the picture also, it's clear that only one oligodendrocyte is there which is forming myelin sheath in multiple neurons. Here only one. This Schwann cell is wrapped for 100 times, which is compacted. I told you the compact protein, which is called as protein 0, and this myelin sheath acts as a insulator. So it prevents it blocks the transmission of impulses. So the impulses are jumps from one node of Ranvier to the other node of Ranvier, and this type of conduction, I told you, it is saltatory conduction. So third type of macroglial cell, which are called as astrocytes. These astrocytes, you can see, these are star-shaped cells. Astrocytes are star-shaped cells, and these astrocytes, they are again two types. So fibrous astrocytes and protoplasmic astrocytes. These astrocytes, what is the function of these astrocytes? They mainly form, here you can see that there are capillaries, and these astrocytes, they form tight junctions in the brain capillaries, and they help in the formation of blood-brain barrier. These astrocytes, they mainly form tight junctions in the brain capillaries, and so that they form the blood-brain barrier. So that is a very important function of this astrocytes. So to summarize the neuroglial cell, you can see the microglial cells, I told you these are only 20%. These are all scavenger cells or phagocytic cells in the central nervous system. What is the function of these microglial cells? They remove debris from the brain. The debris which can be formed from either injury to the brain, or if there is infection, or if there is any degenerative disease which can form the debris, and this debris must be removed from the brain because there is no lymph in the brain. So this debris is removed mainly by the phagocytic cells, and this microglial cells, they resemble like tissue macrophage cell in the body, and one more thing, these microglial cells, they are also formed from macrophages outside the nervous system. And macroglial cells, three different types: oligodendrocytes, Schwann cells, and astrocytes. Astrocytes are two types: fibrous astrocytes and protoplasmic astrocytes. Fibrous astrocytes are more in the white matter, and protoplasmic astrocytes are more in the gray matter, and these are star-shaped cells. So you can see they are star-shaped cells, which are the astrocytes. And what is the function of astrocytes? They form blood-brain barrier in the brain capillaries, and they also produce substances which provide nutrition to the neurons, which are called as neurotropins. Neurotropins are the substances which provide nourishment to the neurons, and these neurotropins are produced from astrocytes. And these astrocytes, they also take up excess hydrogen ions, which helps to maintain the efficiency of the signaling between the neurons. And they also take up neurotransmitter from synapses after release from the neurons, and they also convert glutamate to glutamine by the action of glutamine synthase. So all these are functions of astrocytes. So it forms blood-brain barrier, it releases or it produces neurotropins for the nourishment of neurons. They take up excess potassium ions, they take up neurotransmitters, and they also convert glutamate to glutamine. The next thing we should remember is exoplasmic transport. I told you from the cell body, the neurotransmitters and the proteins are synthesized, and they are transported to the nerve terminals. So the secretory vesicles are also synthesized from the cell body, and they are transmitted to the nerve terminal, where within the secretory vesicle the neurotransmitter will be stored. So there are two different type of exoplasmic transport: orthograde transport and retrograde transport. This orthograde transport, which is also called as anterograde transport, it is two types: fast axonal transport, where the substances are transported 4 millimeters per day, whereas in slow axonal transport, the speed of transport is only 0.5 to 10 millimeter per day. So this is anterograde transport. This anterograde transport is mediated by microtubules, especially by the kinesin. So the kinesin is the microtubule protein which helps in the anterograde transport, and the retrograde transport, from where the from nerve terminal, the empty secretory vesicle, once the neurotransmitter is released, the empty secretory vesicle will be transported again for the filling of the recycling of the neurotransmitters to the cell body, and this transport from the nerve terminal towards the cell body, this type of transport is called as retrograde transport, and this is mediated by a microtubular protein which is called as dynin. So kinin is the protein which helps in the anterograde transport, whereas dynin is the microtubular protein which helps in the transport of retrograde transport. So the speed of retrograde transport is 200 millimeter per day. So fast axoplasmic transport, it is 400 millimeter per day. Slow axonal transport, it is 0.5 to 10 millimeter per day, and in case of retrograde transport, it is 200 millimeter per day. So answer for this MCQs: a patient with multiple sclerosis having low conduction velocity, what may be the possible cause? Loss of multiple sclerosis. So it is with conduction velocity, low conduction velocity. What is the cause? Loss of oligodendrocytes, loss of myelin sheath, defect in node of Ranvier, sodium channel block. So the answer for this is, it is loss of myelin sheath. I told you, when there is destruction of myelin sheath, it leads to a in the autoimmune disease which is called as multiple sclerosis. There will be loss of myelin sheath, which is characterized by, it is associated with conduction block. Node of Ranvier is seen in, where the node of Ranvier will be present? Cell body, dendrites, axon, terminal button. So node of Ranvier is present in the axons. So in the axons, it is surrounded by a myelin sheath. In some places there is no myelin sheath, that is called as node of Ranvier. In a neuron, graded electrogenesis occurs at, so in which part of neuron graded potential is generated? So which is the place? It is the soma dendritic zone. From dendrites, the graded potentials are generated, whereas in initial segment there is propagated action potentials are generated. So initial segment is the site where the action potentials are generated. In what neurons? It is in the spinal motor neurons. In case of cutaneous sensory neurons, first node of Ranvier is the site of propagated action potential generation. Initiation of impulse starts in, from where the initiation of impulse will be start? It is in the initial segment. Initial segment is the site of impulse generated. So that is the action potential. So myelination in PNS is done by, so this is the direct question: astrocytes, oligodendrocytes, squam cell, ependymal cell. So in peripheral nervous system, myelin sheath is formed by squam cell. In oligodendrocytes form myelin sheath in the central nervous system. So astrocytes are the they are macroglial cells which are mainly forming the blood-brain barrier. So pseudo unipolar cells is seen in, so pseudo unipolar type of neurons are seen in invertebrates, retina, spinal motor neuron, dorsal root ganglion. So the answer is dorsal root ganglion. So we know, so invertebrates have unipolar type of neuron, retina and olfactory epithelium will have bipolar type of neurons, spinal motor neuron will have multipolar neurons. So the answer is dorsal root ganglion. Orthodromic conduction. The impulses will be transmitted only in one direction. That is a type of conduction which is called as orthodromic conduction, whereas the axoplasmic transport can be on either ways. So it can be from cell body towards the axon terminal, which is called as anterograde transport, whereas the transport can be from nerve terminal to the cell body, that is called as retrograde transport. So here it is orthodromic conduction. What is the meaning for orthodromic conduction? So read, an axon can conduct impulse only in one direction. So this is the answer. An axon can conduct impulse only in one direction. That is the type of conduction which is called as orthodromic conduction. An axon can conduct impulse in both directions. This is wrong. Jumping of impulse from one node to the next node, it is called as saltatory conduction. So axonal transport is anterograde, retrograde, anterograde and retrograde none. So axonal transport can be both anterograde and retrograde type of transport. Phagocytosis in CNS is done by, so it is by microglial cells. So astrocytes, squam cells and oligodendrocytes, all these are macroglial cells. So the microglial cells are the phagocytic cells in the central nervous system. Thank you.